So good evening or good afternoon or good morning. Thanks for joining the Cambridge Genomics Meetup. So as you know, this is a group of scientists, students and professionals who have decided to come together into some kind of community around Cambridge. But of course, we're not focused on the Cambridge only set of scientists, as you can see from the people who are joining us today. So this time we're going to be talking about genome assembly. I want to say that actually we have a meeting at the same time, 7 p.m. British Summer Time in this case, to 9, every second last Wednesday of the month, second last Wednesday of the month. So the next month we're going to have another meeting on the 20th of May, which is Wednesday as well, and it'll be on medical genomics. So I hope that many of you have the opportunity to join us. And now Sina is going to introduce our first speaker.
Yes, so our first speaker is also from our sponsor, Dovetail Genomics. It's Kai Kleiber. And Dovetail Genomics specializes, amongst other things, in assembly or finishing of complex eukaryotic genomes. And they have assembled things we probably all like oats or lettuce to high completeness, but unfortunately also a few that we never want to meet, like mosquitoes or white sharks. Well, right now we're unlikely to meet them. And he's going to explain to us how they do that and give us an introduction to genome assembly.
Yeah, thank you. First of all, we have a lot of mosquitoes in our garden right now, so that is not entirely true. But yeah, so maybe a bit more to Dovetail Genomics. So it started as a genome assembly service company, and recently we moved into kit sales as well. There is this one kit we are calling OmniC, which is basically Hi-C, which is what I'm going to mainly talk about. And OmniC is just a very marketing-y, fancy name for a certain variety of Hi-C. But what I'm going to focus on is how you scaffold a genome with Hi-C. But as introduction first, genomes are assembled for a really long time. So obviously you started with very small genomes in the very beginning. And about 20 years ago, we had the first assemblies from a human genome and from mouse. And actually these assemblies are not even that bad, mainly because Sanger was used. Sanger has comparatively long reads compared to next generation sequencing, over 1,000 kilobases. Base pairs, sorry. And assembling this is a bit easier. Then Illumina came, sequencing got really, really cheap, but read lengths got shorter as well. So it got more difficult to assemble these. So you can see here that from now up until, well, this is a little older, so up until 5 years ago, we got more and more assembled genomes, but actually the contiguity, so which is measured by the scaffold N50 in this case, actually went down a bit. It went up now because now you have all these long read technologies like PacBio or Nanopore, which contribute to continuity of the genome as well. So this is, if you assemble a genome, this is how it more or less looks like. So when you have mostly short reads. Illumina, for example, usually 300 base pairs. PacBio has about 15 kilobase reads, so they're significantly longer already. Nanopore can go up to megabases, up to a certain degree, but you still have to assemble this. So just an example, mammalian genome has 3.3 gigabases, and you can see if you only have 300 base pair bits and pieces, it's really hard to put them all together. So you're really dependent on the overlap in between. If you have 15 kilobase reach, it's significantly easier, mainly because you have way more overlap compared to Illumina reads. But still, even if you put these together, you get context. So these are basically now consisting of these short reads, but there are gaps in between. And you have to bridge these gaps, and there are several ways to do that. Traditionally, for example, mate pairs were used for this, then 10x. If you know the company 10x with the chromium, they had a way to do that as well. And Hi-C was another technology that can connect these contexts and make a more contiguous genome. This is basically how it looks like, more or less. So different in different scales, the genome assembly works. So shotgun usually has hundreds of base pairs. Mate pairs goes up to 10. Optical maps is like BioNano, for example, can go to hundreds of kilobases. PacBio works in the 20 kilobase range. 10x would work in a similar range as optical mapping. And then high C actually goes up to chromosome scale. It has advantages and disadvantages, and I will get into detail at that. But because Dovetail is a high C company, or generally a proximity ligation company, I'm going to focus on high C and how this works in genome assembly. So first of all, high C is a method. It's basically a DNA library prep method. And what you do in this case is you start with tissue. So you do not extract DNA first. You just start with blood, with muscle tissue, any tissue you like. You flash freeze it, and you cross-link everything from aldehyde. So basically what happens then is these are histones. This is DNA. And you lock the chromosome in 3D space. So 1 and 2, for example, at two distal ends of the DNA, they might be in very close proximity in 3D space. But if you look at linearly, they're very far away. And traditionally, if you've ever seen a DNA sequence, you read it like a sentence. So it starts at one end, it goes to the other one, and then there might be thousands of base pairs apart, but in reality, they might be next to each other, like here. So first, you cross-link everything with formaldehyde, so you fixate the chromatin. Then you digest it with the restriction enzyme. You get these sticky ends here. You biotin label these sticky ends, and that's the important part. Now you ligate everything back together in close proximity. So that means 1 and 2 at distal ends of the piece of DNA are now ligated together. So after that, you reverse cross-linking, and you do a pull-down with streptavidin beads for these biotin labels. And then you're getting basically an Illumina sequencing read. So that means read 1 and read 2 are now at two distal, are on the same fragment. But if you're mapping this back to the genome, it's actually at two different locations. And I have a slide for this in a second. So this is traditional high C. If you see a paper that used high C, most likely they use this method. Dovetail recently launched a new kit, and high C works slightly different here. You still have the same input material. You still cross-link your DNA, but you're digesting now with DNAs instead of a restriction enzyme site. So as you know, restriction enzymes, they only cut at certain motifs, while DNAs cut uniformly across the genome. It's motif independent. So after that, and so instead of religating these ends directly, we are ligating a bridge adapter that is biotin labeled in between. But after that, it's exactly the same. So you remove the cross-linking, you clean it up, you do a pull down, and you're getting these Illumina reads where read 1 and read 2 map back to different locations on the genome. Just on a side note, it's a 2-day protocol, 1 day for the high C part, 2nd day is just a DNA library prep. It's fast, because it's a kit, it's faster. There's a homebrew version, which usually takes up to 4 to 5 days. This one takes 2 days. And for the people who have ever done that before, there is no fragmentation needed, so you do not need a Cuvaris on the 2nd day. The insert size is already correct. Yeah, but now to look at these sequencing reads you get. So as I mentioned, this is now the fragment you have seen before. So read 1. So this would be the read. This is a reference genome. So let's say if you have human data, humans have a very nice reference genome with HC38, for example, and you take all your high C reads and map it against it, and you can see read 1 maps back here, and read 2 maps back here. Statistically, of course, you're going to have way more reads that are in close proximity to each other than in large proximity to each other. So just so you know, this is read pair separation. So the higher this dot is, the further apart read 1 and read 2 would map back on a reference genome. So this is how it would look in reality. So again, every of this dot you see here is a read pair. And always think in triangle if you see these. So this is a contact matrix. So this one maps back at this location, at this location. And these are two chromosomes. And you can see, because you lock the chromatin in 3D space, you're basically binning the data within the chromosome. So all the reads are here within chromosome A, all the reads are within chromosome B. There are a few reads in between. These are called transreads. They're mostly ignored. They actually have some interesting information as well. So while these reads in the chromosome will tell you the 3D structure of the chromatin, so how it's folded, the cis read, the trans read will tell you how chromosomes are located next to each other. So there was a very nice talk I recently saw where they showed that, for example, highly transcribed chromosomes are closer to the nuclear membrane, which makes sense because then the RNA doesn't have to travel that far outside. And these, you can see it with these reads. I honestly don't know how. I'm not a bioinformatician, but apparently you can do that. So, okay, what's now the difference between traditional high-C with restriction enzymes and high-C with DNA? So why would you do DNAs? And the reason for that is coverage. So this is an example of high-C data plotted back against the human reference genome. And you can see here, this is with one restriction enzyme site. There is a high-C protocol with several restriction enzyme sites, and you can see here, there are gaps. This is similar to what I showed to the very beginning for the people who do not know genome assembly that well. You can see all these gray stuff are Illumina reads mapping back to the reference, and you can see there are gaps in between here. And this is mainly here because there's no restriction enzyme site. And because there's no cut, there is no data. So that's why this is not complete. Because we're using DNAs here, there is a significantly more uniform coverage across the genome. Just to compare this to shotgun, which is the go-to method for doing whole genome sequencing, the dotted line here is a distribution of shotgun library, and the blue line here is a distribution of the DNase-based high-C library. And you can see the distribution for restriction enzyme base is pretty much all over the place. You can use this for contract matrices as well. So this is similar to this one, more dense data. So all the orange stuff are high-C reads. The white lines you see here are basically sites without restriction enzyme sites, so that's why there is no data, and that's why you can't see it. Side note, these are TAT structures. So this is these triangles you see here everywhere show you how DNA is folded. So if you're looking, can I actually just zoom in? Let me try this. Okay, it works to a certain degree. So you see the small dot here on top of it. So you know that this part of the DNA and this part of the DNA actually make a loop, and they touch here. So this is what you can see in this data. Yeah, this is high-C data. If you use the DNA-based approach, you can see here you do not get as many of these white lines, so you get more crisp data, so to say. If you zoom in, it's a little bit, it's even more pronounced. Here you can see way more gaps. If you go to a much further resolution, so you amplify this, you can see even way more gaps here. For people who work on cancer, TERT is here, which is a quite important gene in cancer. So you can resolve this better with OmniC. Also, on a side note, what I just said, loop calling, because we just spent more data. So this is, for example, a piece of DNA. Actually, there's a promoter, there's an enhancer region, there's a 130 kilobase difference in between. You can see here the enhancer region is recruited to the promoter. This is actually the SOX2 gene, which gets expressed. And this dot here basically show you that this part and this part, or if you think of triangles again, this part of the DNA and this part of the DNA are next to each other. So high-C data shows you that. Good. Okay, this was a lot of epigenetics, but how does this help in genome assembly? And this is about genome assembly. So if, let's say, you have your, you sequenced your genome of interest, you do not have a reference, you will get these contexts here. So that means parts that you can assemble, but there's going to be gaps in between, and you have to put them somehow together. What you do now is you create a high-C, so you have your reference. So it's PacBio, Illumina, 10x, however you wanted to create it. So now you create your high-C data from exactly the same individual, and you map it back against the reference genome you made. And then, so this is again the same thing. So every of this dot is a read pair. Think in triangles again, mapping back at 2 different locations. And as I said, statistically, you're going to have way more reads down here. So in close proximity, only 10 kb apart, than up here, that means 10 megabases apart. So if you map this back and suddenly you get this cloud of reads up here in this bow tie structure, statistically, this should not happen. So you have a software, at least our software called HiRise. You're scanning all these reads here, and suddenly you see this. Then you know something is not correct here. So this contig, there's something wrong with this. So it sees that. So it takes this contig and switches the orientation around, and all these reads fall back into these gaps you have here. So it corrected the orientation of that contig. And this is how you correct. You can use Hi-C data to correct the order and orientation of the context of your draft genome in the first place to make it more contiguous. Side note, again, if you plot this data against the reference genome, so let's say you create a Hi-C library from your personal genome and compare it to a reference, and you get something like this, that's actually a biological effect. So that means you have a inversion in your DNA, or at least compared to the reference. So Hi-C can actually show you that. So if you have structural variation in cancer, Hi-C will show you this in these patterns here. Good. But back to genome assembly. So traditionally, if you want to use a, if you're scaffolding with Hi-C, you need an input assembly of 1 megabase and then 50 of 1 megabase. I don't know how many people know what N50 actually means, but just to make it very short, it gives you an idea of the average context size of the genome. I should have actually put a slide to explain that. But okay, just go for an average context size. 1 megabase is quite high. So for example, if you have a human genome and you do an Illumina assembly, you get a N50 of 20 to 80 kilobases. So very, very low. If you go with PacBio, you get into the megabase range. And if you go with Nanopore as well. So that's why if you, for example, have a plant, they're very repetitive, very heterozygous. They're significantly harder. So even the genome size might be smaller, only 500 megabase compared to a human with 3 gigabases. The assembly is significantly harder because you don't have a reference, and it's very heterozygous, and it can be very repetitive. Anyway, so this is an example actually for a mammal. This is a sea lion. The input assembly was 26 megabases, which is very, very good. And Hi-C scaffolding went up to chromosome level. So in this case, you have from 26 megabases, you went to 140 megabases. This works well. Any kind of Hi-C works here. However, this is now a different example. This is grass, so a plant, significantly smaller genome with 600 megabases. This was a PacBio assembly, so with long reads, with an input of 50 or 380 kilobases. And if you use Hi-C only, with such a fragmented genome, you only get to 5 megabases. This is not chromosome level. So in this case, you need an intermediate scaffolding step, and Dovetail has one called Chicago, which, without going into too much detail, is an in vitro version of Hi-C, using high molecular weight DNA as input material instead of tissue. So back then, what we did is we took this assembly, we did an intermediate scaffolding step, so improved it from 380 to about 800 kilobases. And then with Hi-C, it went up to chromosome level, about 45 megabases. Yeah, so this is the traditional thing, how we did things. This is with restriction enzyme-based Hi-C. However, now we have the, so how does DNase-based Hi-C play into it? So to visualize how you scaffold a contig, you need information to do so. So that means you can only scaffold a contig if you have a restriction enzyme site on it, because only then you have data. So that's why you need an N50 of a megabase, because then statistically the contigs are large enough that you will have at least one restriction enzyme site on it. if you use OmniC, because you're using DNAs and you're not dependent on the cut motif, statistically, you have a cut site even on significantly smaller contexts. So what does this mean? It means you can scaffold much more fragmented assemblies better. I'm not going to go into too much detail. So these are just actual assemblies we did. These are quite high input assemblies, 27 megabase, 10 megabase, 2 megabases, Chicago and high C. So high C and OmniC, there's no difference. So it all goes up to chromosome level. It's more interesting for more fragmented input assembly. So this one has an input assembly of only 670 kilobases, 400 kilobases, 140 kilobases. This is a snail, and if anyone ever assembled a snail, a mussel, they're super hard. They're very repetitive. This is a hybrid assembly using PacBio and Illumina data with an input assembly of 200 kilobases. And OmniC alone was able to scaffold all of these up to chromosome level in one scaffolding step. So this one went from 200 kilobases to 145 megabases directly. We even have two data sets which you should take with a grain of salt, but we actually created them. This is a mammal, a hamster, Illumina only assembly with an input N50 of 80 kilobases, and OmniC alone, without any intermediate scaffolding step, went directly to 127 megabases, which is a lot. The same thing for an amphib. This is a fairly large genome, 4.2 gigabases. Just for the ones who don't know, human has 3. This one had an input assembly of 5 kilobases, and it scaffolded directly to 140 megabases in one step. Having said that, if you would come to me or to Dovetail as a company and you would say, you have this input assembly, I would tell you, do not do this. And I'm very sure if you look in a bit more detail here, there is going to be on a smaller scale, like very small context, still in the wrong orientation, but generally this is something you could do. I would never recommend this. You should really not do this unless you have no other way. And there are some examples. For example, we had a protected shark. There was only very limited sample. So they just had an Illumina assembly they had to work with, and they couldn't get any PacBio reads. PacBio has the disadvantage that you need a lot of input material. So they had to do this. Yeah. So because of the properties of OmniC, what you can use this library for is for genotyping as well. Because here you can see these are this is a software called GATK, which you can use for SNP calling. And this is a comparison between shotgun, OmniC, and restriction enzyme-based IC. And just to make it short, you almost get all SNPs that you get with shotgun as well. There's a significant gap to restriction enzyme-based because you have, again, the cut bias, but you get most of it. That has one huge advantage, which plays into assembly as well. You can phase the genome quite well. I'm not entirely sure how much people know about phasing, but let's put it that way. Human is diploid, so you have one copy from your mom and one copy from your dad. There are differences between both. Most of the time, when you look at an assembled genome, you collapse everything into a pseudo-haploid, meaning you don't look at both mom and dad separately. You look at a combination between both. But you can take this apart using high-C data and a software called HapCut2. And this is how you can phase a genome, and then you can actually look at both phases separately. You can even do this with polyploids. Honestly, no one has tried this yet. This is a paper from middle of last year where it's a pipeline called All High C. So this should allow you to phase even a polyploid using high C data. So I've been told the OmniC data should be compatible with this data set. There are 3 groups right now trying this on tetraploids, but I haven't seen any data back because I said the kit is fairly new. Okay, that's already it. If you have any questions, you can either, should we do it at the end, or if anyone has questions now, they can ask me as well.
There are a few minutes, so if anyone would like to ask any question. Okay, so I'm gonna ask the first question. Tell us a bit about how does Dovetail works? How do you make money?
So if you want to use it as a service, you can send us a piece of tissue, and we would sequence that with Illumina or PacBio, most likely PacBio. It got significantly cheaper recently. Then we would create these scaffolding libraries that I basically showed you here, the OmniC libraries. And then we have a software called HiRise, which is proprietary to Dovetail to make these assemblies. So the statistics I showed you here, for example, were created with that. The other thing that we are doing is we're selling kits now. So this OmniC you saw here, you can just buy the kit and create these libraries yourself. And there are freeware tools there as well. So Salsa 2, for example, is one of them, or 3D DNA. And then you can just take an Hi-C data set and scaffold your own genome, which of course is significantly cheaper.
Great. So I guess if anyone would like to use Dovetail services, they need to send you an email.
Yeah, or if they want to have the kit, I have actually a whole other talk about epigenetics, which is what people mostly do nowadays with Hi-C.
Okay.
Yeah, but we can do this another time. I know this was a lot, and Hi-C is fairly hard to understand, especially the bioinformatics part. So if someone has like in-depth question, just reach out to me.
Great. So there'll be more opportunities for asking questions, Kai.